In regards to the recent developments of COVID 2019 and the authentication of WHO interfering into collaborating with the scientific researchers of the Chinese institutions on the formulations on the origins of COVID 2019 is to stipulate any further aggression or scientific evidence to project that efficaciously that the COVID 2019 was not subjected in forms of the leak of a lab, because there's enough substantiating evidence on the analysis of the COVID 2019 on the genetic strain that of the 29,000 genomic structure of the S spike protein and the M protein in the layers inside the COVID 2019, the genetic stored information, is that through the analysis there's some conjecture that association with if bats on which was analysed by the Chinese university and institution on the genetic strands in which they found genomic sequence, even though they have found deficiency viruses a bit like similar to exosomes, where it contains genetic information over variations of the nucleotides, which includes guanine, cytosine, and RNA is made up of uracil, and thymine is DNA. So DNA is a double-stranded RNA, basically, where RNA is the deoxynucleotide made up of a long chain of nucleotides, which is made up of purines and pyrimidines, which include thymine, guanine, cytosine, and these are associated with adenosine triphosphorylation, guanosine triphosphorylation, where triphosphorylation is the addition of a phosphate group, on which there's enough information to suggest that from the analysis of researchers that it did not leak from it was not leaked from a genetically engineered, from a biologically chemical engineered weapon, or it came from a lab. There's not enough substantial information to say that it came from zoonotic behavior. Zoonotic is the host being able to find a reservoir through mammal to human transmission. So the mammal is associated with bats, on which from the novel coronavirus, from the tree diagram of the association from the history, that came from bats, as it was 99.9 over exceeding the majority, 99.99% effect associated with bats of the COVID 2019 strain. So there's enough evidence to say that because due to the host being able to find a reservoir due to something called Charles Darwin theory, where through evolution through species of genetic offspring, they inherit characteristics allowing the propagation of the virus. So when the human comes in contact with a mammal species, which is still unknown, but it came from either pangolins, bats, or another unknown source of information, but there's enough evidence to suggest, due to radioactivity and radioactive decay of exposure to radiation, allows there's something called a mutation. Mutation is just the displacement of a elongation of a replacement of the amino acids along one of its nucleotides, basically. So it's not similar to what you see in aliens of basically mutation. It's just a bit like isotopic decay, where isotope is the nuclei decay of isotope of the mass, where it turns protons. So basically, when through replication of the virus through human activity and the cytoplasm, through transcription and translation, it allows the mutation to occur, where it changes from basically maybe from R to Y. It's like adenine to alanine, or a phenylalic acid, or lysine, or guanine. So basically, it's similar to that association, basically. So during that process, now this allowed this so the because of due to the viral replication and how it's there's an endemic and a pandemic at the same time in global transmission of something known as the B117 strain, which is associated with the immune response. So basically, the virus has replicated through something known as Charles Darwin theory, where it's evolved to an extent that it's evading the immune response due to so much mutation because of things like restrictions from quarantine or restriction from travel, less restrictions from travel, where humans congregate in spaces of close proximity of a high entropy, where of of a high entropy, where it allows individuals to asystematically transmit the virus to the individual. So the B117 strain is just associated with the immune response, where it's restricted from things like acute myeloid leukemia and things like actimira, which reduces inflammation in the body because it invades the immune system, so it affects the individual host cell at the same time. So because there's a large continuum of the B117 strain, there's a lot in other B117 strain. It's evading the immune response, where the attacks the ACE more easily, but due to what I believe hypothesis is increase increase in anxiety and high sodium intake increases because there's information. Okay, even though there's information that there's a high association with collagen TMPRSS2 and the ACE2 angiotensin converting enzyme associated with angiotensinogen converting ACE127 to ACE. So associated with upregulations in the renal category, because ACE is just a sync complex ion with carbox peptidase. So it's a sync transition element attached with formations of proteins of the amino acids, and basically, so if you elevate the sodium level, and if you even increase the anxiety, because due to this COVID 2019 is having a negative impact on the individual's mentality, increase the anxiety, so it increases anxiety and adrenaline levels, association with increasing stress levels and cortisol levels, um, and increase in adrenaline, and increase in BP is blood pressure. Blood pressure allows the hemoglobin to transfer the oxygen to the body. So if you are restricting the blood capillaries, so this is associated with vasoconstriction and vasodilation from prostaglandin release. So increases in sodium levels, basically it increases. So when we have anxiety levels, and the renal kidneys releases sodium, releases sodium from sodium retention. So it releases sodium levels, which increases the ACE distribution. So it increases the possibilities and the probability of the propagation of the virus into the endothelium of the outer cell through endocytosis in viral entry. So basically, our anxiety level is associated, but due to exposure from the media, and it increases our cortisol, anxiety levels, and adrenaline, which is associated with increasing sodium levels. Sodium levels is associated with ACE2 attacking more easily, attacking the endothelium of the cell. So if we control this, and evading the immune response due to transcription translation change. So transcription is the copying of the 3' the 5' through elongation elevator, basically, of the RNA, which is RNA dependent polymerization. They are in antiviral treatment at the minute, but due to the vaccine is just the vaccine, for example, AstraZeneca, is taken from chimpanzees of the genomic sequence of a human, which causes the injected inside the body to allow the body to initiate and recognize it. So basically, but the thing is, due to the adaptation of the virus in a global pandemic at the minute, where in different countries there are different variations of the COVID 2019 antigen associated with that evades the immune response, from the B117 to the M501Y and the 487K, the Brazilian variant and the Nottingham variant and the UK variant and the African variant. But these are all the same strain, all the same strain from the same denoted from the same, like a subgroup of the family, like a subgroup of the family. But due to genetic response and genetic mutation of the displacement of the RNA, so basically this reduces the chance of the binding of the antibodies, because the RNA makes the mRNA cause the human adenovirus, which is injected inside and made to produce the antibodies response, because it causes the messenger RNA to create a spike protein similar to the authentic strain of the original strain of COVID 2019. But due to something called of an immune response evasion due to genetic mutation, it does not allow the epitope, the antibody to of the hinges of the hinges to attach more easily, to neutralize the S spike protein to prevent viral entry or block viral replication. So these vaccines are efficacious, and there are things like there has been media exposure, due to large media exposure, that this COVID 2019 causes blood clots, but blood clots is caused by over-inflammatory cytokine storm, where basically when the body has viral no, has a foreign viral element inside our body causes an innate adaptive immune response that attacks the virus. But due to overstimulation from the virus, this is caused by over-inflammatory response, which attacks the neurons inside our cells, and it attaches our nervous system, our cardiotic response of pneumocytes and phagocytes inside our body, which is associated with carrying oxygen, so it causes things like blood clots. But this can be suppressed by instigation of non-steroidal anti-inflammatories like ibuprofen or aspirin or blood thinners. Blood thinners reduces thrombosis and blocks something known as COX-1-2, which blocks cycloogenesis 1-2 from andronic acid, that blocks platelets aggregation associated with apoptosis and platelets, and EGCF control factor, which basically, when it blocks it causes prostaglandin release, and it causes the capillaries to widen, which causes vasodilation, which vasoconstriction, vasodilation is the widening of the blood capillaries, and blocks platelets aggregation from PGSX1 and PGX2 at the same time, associated with EGCF. And this is associated with something known as oxidative stress, due to something known as van Willebrand factor at the same time. So this is all associated with we can introduce things like hypodensity lipid, like high density lipoproteins, which increases oxidative stress due to increasing cholesterol and blocking of the blood capillaries, basically. So these are blood clots because from some reason the virus has the capability or intellectual capability of invading and interacting with the neurons inside our body, as well as our cardiac cells inside our bodies. So basically the intervention is known as aspirin or ibuprofen, prevents the blocks something known as COX-1, because this bromophene and in the endocrine system it affects an endocrine and associated with thrombosis and platelets aggregation and blood clots, which is in the cardiac arrest, and basically hypopenia as well, and oxypenia at the same time of from thrombosis. So this increases vasodilation, which so blood thinners, like for example chocolate is a known dark chocolate can be utilized as a blood thinner. So even these vaccines can show some form of efficacious, but it does not guarantee the evasion of increase in transmission of increase of R0, where R0 is the number of second cases per primary in a total susceptible population, where R is the transmission parameter, gamma is the exiting the infected class, which includes gamma, mu, and nu, where gamma is the infected class, the recovery rate, and mu is the birth of infections, and nu is the death, which is the mortality. So this does not associate. Okay, there has been that media exposure that increases. There has been that the different age groups are susceptible to the vaccines, which includes AstraZeneca, Pfizer, Novak, the Sputnik vaccine, and other Israeli vaccine, and other antiviral remedies, or the antibody cocktails. These are associated with decrease in susceptibility, because there would be an increase in the population as there'd been an increase in individuals having these antibodies. The antibodies created from the intelligence of scientists. But does not guarantee that vaccines will affect, but vaccines with other antiviral treatments and more methodologies on understanding and analysis through analysis and approach of antiviral drugs implemented that can prevent, like hydroxychloroquine, remdesivir, sonamifir, acyclovir, HIV medication, malarial medications, and other medications, or TMPRS2 or serotonin protease, or CRP glucose regulated proteins inhibitors, and that kind of thing. So this is the TMPRS2 inhibitor, prevents viral entry of upregulations. So increases in lysosomal pH, reduces viral release in the in the lysosome into the cytoplasm for transcription, which releases into transcription, or in the ribosome at the same time of the formulations of the DNA associated with the compilations of the 20 amino acids associated with. So understanding more about this allows us to understand, so understanding the interventions of how the viral gains entry, and how things like, for example, H1N1 uses something called as N-acyl numeric acid, which is blocked from sonamifir and Relenza, basically, because this shows the drug structure of the antiviral is associated with N-numeric acid, but this is a different form of virus, basically, which includes, because it attaches the cell, for example, the H1N1 attaches through hemagglutinin glycoprotein, and leads through the cleavage of the neuraminidase glycoprotein through a cleavage. But understanding this, like for viral entry, scientists do understand viral entry attaches from my analysis that the ACE attaches to the ACE. The ACE2, as well as affects the TMPRS2 regulations of coliform for viral entry for endocytosis. There's something known as a conformality change from pH. So if we increase our pH to acidity, basically, acidity is the number of oxonium ions in our body. If we increase it, it causes something called a conformal change, of which the S-pipe protein that attaches to the ACE changes structure in a way it's adapted to increase in viral entry. But there are things that block viral entry, like hydroxychloroquine prevents by increasing the lysosomal pH of the alkalinity in the body, and prevents RNRDP from occurring, which is RNRDP dependent polymerase, ribonucleic acid dependent polymerase through the transcription phase. But they are that antiviral remedies don't happen in general, because they are not utilizing known as zinc, and because the hydroxychloroquine acts like an ionophore, and the ionophore allows the the zinc allows the zinc to be attached to extracellularly enter intracellularly to block RNRDP, but just utilizing like chloroquine or hydroxychloroquine will not effect efficaciously. But we've known some form of zinc inside intracellular zinc inside our bodies that blocks RNRDP and blocks viral replications. But things like remdesivir at the same time that blocks is a similar nucleotide to adenosine or guanine triphosphorylation, which acts similar to that nucleotide, and it blocks viral replication at the same time through transcription phase of the promoters or primers to free primers at the same time. So there are things like that that are being utilized and utilized as well, or things like ibuprofen prevents inflammation, or the introductions of Actimera or tocilizumab, which is a monoclonal antibody that prevents BCL kinase from occurring inside, and the platelet aggregation and platelet aggregation basically, and the signalations of the STAT as well. So these prevent pro-inflammatory response by blocking something of BCL kinase. So there are lots of things that scientists are working on, but utilizing things like vaccines in general will suppress the propagations of virus. But understanding more about Actimera and tocilizumab has found to be reduced hospitalization by a large proportion and as well. So understanding this with antiviral treatments and other vaccines, and vaccines are just mRNA creating the spike protein to allow our body to adapt to it, to create the to create the antibodies through mRNA to destroy the spike protein from neutralizations antibodies at the same time. So understanding this, like, and understanding the deployment of different various efficacious vaccines like AstraZeneca, Pfizer AstraZeneca, and Oxford AstraZeneca vaccine, the BioNTech, the Sinovac, and working collaboratively, where scientists are working collaboratively in 24 hours a day to find the answers to COVID-2 2019. So in the future, this will suppress viruses and reduces R0 and R0 the rate of transmission, as there have been a larger proportion of individuals with the antibodies inside. Thank you.